This video tutorial is all about the transport of oxygen. Oxygen is transported in the red blood cells, which are known as erythrocytes. These contain the protein hemoglobin. When hemoglobin binds to oxygen or takes up oxygen, it becomes oxyhemoglobin. Hemoglobin is a complex protein with 4 subunits. Each subunit consists of a polypeptide chain and a heme group, which is a non-protein group. The heme group contains a single iron in the form of Fe2+. This iron can attract and hold an oxygen molecule. The heme group is said to have a high affinity, which is another way of saying attraction, for oxygen. As each heme group can hold just one oxygen molecule, each hemoglobin molecule can carry 4 oxygen molecules. It has been calculated that there are about 280 million molecules of hemoglobin in each and every red blood cell. So a red blood cell can carry over 1 billion oxygen molecules. Oxygen is absorbed into the blood as it passes the alveoli in the lungs. Oxygen molecules diffusing into the blood plasma enter the red blood cells. Here, they become associated with the hemoglobin. This means that the oxygen binds reversibly to the hemoglobin. This takes the oxygen molecules out of solution and so maintains a steep concentration gradient, allowing more oxygen to enter the blood from the lungs and diffuse into the cells. The blood carries the oxygen from the lungs back to the heart before travelling around the body to supply the tissues. In the body tissues, cells need oxygen for aerobic respiration. Therefore, the oxyhemoglobin must be able to release the oxygen. This is called dissociation. The ability of hemoglobin to associate with and release oxygen depends on the concentration of oxygen in the surrounding tissues. The concentration of oxygen is measured by the relative pressure that it contributes to a mixture of gases. This is called the partial pressure of oxygen, or symbolized as a little p followed by O2. It is also called the oxygen tension, and it's measured in the units of pressure, kPa. With a normal liquid, you might expect the concentration of oxygen absorbed into the liquid to be directly proportional to the oxygen tension in the surrounding air. A graph of potential or percentage saturation plotted against oxygen tension would be a straight line, it'd be linear. This is not the case with blood containing hemoglobin. Hemoglobin can associate with oxygen in a way that produces an S-shaped curve, like the one shown on the slide in front of you. This is called the hemoglobin dissociation curve. At low oxygen tension, the hemoglobin does not readily associate with oxygen molecules. This is because the heme groups that attract the oxygen are in the center of the hemoglobin molecule. This makes it difficult for the oxygen molecule to reach the heme group and associate with it. This difficulty in combining with the first oxygen molecule accounts for the low saturation level of hemoglobin at low oxygen tensions. As the oxygen tension rises, the diffusion gradient into the hemoglobin molecule increases. Eventually, one oxygen molecule enters the hemoglobin molecule and associates with one of the heme groups. This causes a slight change in the shape of the hemoglobin molecule, known as a conformational change. It allows more oxygen molecules to enter the hemoglobin molecule and associate with the other heme groups relatively easily. This accounts for the steepness of the curve as the oxygen tension rises. As the hemoglobin approaches 100% saturation, the curve levels off, creating the S-shaped curve. Mammalian hemoglobin is well adapted to transporting oxygen to the tissues of the mammal. The oxygen tension found in the lungs is sufficient to produce close to 100% saturation. The oxygen tension in the respiring tissues is sufficiently low to cause oxygen to dissociate readily from the oxyhemoglobin. Fetal hemoglobin is slightly different from adult hemoglobin. Fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. Therefore, the hemoglobin dissociation curve for fetal hemoglobin is to the left of the curve for the adult hemoglobin. This is because fetal hemoglobin must be able to associate with oxygen in an environment where the oxygen tension is low enough to make adult hemoglobin release its oxygen. In the placenta, where the oxygen tension is low, fetal hemoglobin will absorb oxygen from the surrounding fluid. This reduces the oxygen tension even further. As a result, oxygen diffuses from the mother's blood fluid into the placenta. This reduces the oxygen tension within the mother's blood, which in turn makes the maternal hemoglobin release more oxygen, i.e. dissociate.